The Na plus K plus pump is driven by the transient addition of a phosphate group. The Na plus K plus pump provides a beautiful illustration of how a protein couples one reaction to another, following the principles discussed in Chapter 3. The pump works in a cycle, as illustrated schematically in Figure 1211. Na plus binds to the pump at sites exposed inside the cell. Stage 1. Activating the ATPase activity. ATP is split with the release of ADP and the transfer of a phosphate group into a high-energy linkage to the pump itself. That is, the pump phosphorylates itself. Stage 2. Phosphorylation causes the pump to switch its conformation so as to release Na plus at the exterior surface of the cell, and at the same time to expose a binding site for K plus at the same surface. Stage 3. The binding of extracellular K plus, stage 4, triggers the removal of the phosphate group, stage 5, causing the pump to switch back to its original conformation, discharging the K plus into the cell interior. Stage 6. The whole cycle, which takes about 10 milliseconds, can then be repeated. Each step in the cycle depends on the one before, so that if any of the individual steps is prevented from occurring, all the functions of the pump are halted. This tight coupling ensures that the pump operates only when the appropriate ions are available to be transported, thereby avoiding useless ATP hydrolysis. The Na plus K plus pump helps maintain the osmotic balance of animal cells. The plasma membrane is permeable to water, see Figure 12.2. And if the total concentration of solutes is low on one side of the membrane and high on the other, water will tend to move across it until the solute concentrations are equal. The movement of water from a region of low solute concentration, high water concentration, to a region of high solute concentration, low water concentration, is called osmosis. Cells contain specialized water channels, called aquaporins, in their plasma membrane that facilitate this flow. The driving force for the water movement is equivalent to a difference in water pressure and is called the osmotic pressure. In the absence of any counteracting pressure, the osmotic movement of water into a cell will cause it to swell. Figure 12.12. In the tissues of the animal body, cells are bathed by a fluid that is rich in solutes, especially Na plus and Cl minus. This balances the concentration of organic and inorganic solutes confined inside the cell. But the osmotic balance is always in danger of being upset, as the external solutes are constantly leaking into the cell down their individual electrochemical gradients. Animal cells thus have to do continuous work, pumping out unwanted solutes to maintain the osmotic equilibrium. Figure 12.13a. This function is performed mainly by the Na plus K plus pump, which pumps out the Na plus that leaks in. At the same time, the Na plus K plus pump helps to maintain a membrane potential, as we explain later. This membrane potential tends to prevent the entry of Cl-, which is negatively charged and would need to move against the electrical gradient generated by the pump to enter the cell. Different cells cope with osmotic challenges in different ways. Plant cells are prevented from swelling by their tough cell walls and so can tolerate a large osmotic difference across their plasma membrane. Figure 12.13b. The cell wall exerts a counteracting pressure that tends to balance the osmotic pressure created by the solutes in the cell, and thereby limits the movement of water into the cell. Osmosis, together with the active transport of ions into the cell, result in a Turgor pressure that keeps plant cells distended with water, with their cell wall tense. Thus, plant cells are like footballs, in which a leather outer case is held taut by the pressure in the pumped-up rubber bladder inside. The cell wall acts like the leather outer case, and the plasma membrane acts like the rubber bladder. The Turgor pressure serves various functions. It holds plant stems rigid and leaves extended. It also plays a part in regulating gas exchange through the stomata, the microscopic mouths in the surface of a leaf. These pores are opened and closed by the guard cells that surround them. Figure 1214. Guard cells control their own Turgor pressure by regulating the movement of K+ across their plasma membranes. In some protozoans living in fresh water, such as amoebae, the excess water that continuously flows into the cell by osmosis is collected in contractile vacuoles that periodically discharge their contents to the exterior. Figure 1213c. The cell first allows the vacuole to fill with a solution rich in solutes, which causes water to follow by osmosis. The cell then retrieves the solutes by actively pumping them back into the cytosol before emptying the vacuole to the exterior. Intracellular Ca2+ concentrations are kept low by Ca2+ pumps. Ca2+, like Na+, is also kept at a low concentration in the cytosol compared with its concentration in the extracellular fluid, but it is much less plentiful than Na+, both inside and outside cells. The movement of Ca2+ across cell membranes, however, is crucially important because Ca2+ can bind tightly to a variety of proteins in the cell, altering their activities. An influx of Ca2+ into the cytosol through Ca2+ channels, for example, is often used as a signal to trigger other intracellular events, such as the secretion of signal molecules and the contraction of muscle cells. The lower the background concentration of free Ca2+ in the cytosol, the more sensitive the cell is to an increase in cytosolic Ca2+. Thus, eukaryotic cells in general maintain very low concentrations of free Ca2+ in their cytosol, about 10 to the -4 millimoles, in the face of very much higher extracellular Ca2+ concentrations, typically 1 to 2 millimolar. This huge concentration difference is achieved mainly by means of ATP-driven Ca2+ pumps in both the plasma membrane and the endoplasmic reticulum membrane, which actively pump Ca2+ out of the cytosol. Like the Na+K+ pump, the Ca2+ pump is an ATPase that is phosphorylated and dephosphorylated during its pumping cycle. Figure 1215. It is thought to work in much the same way as depicted for the Na+K+ pump in Figure 1211, except that it returns to its original conformation without binding and transporting a second ion. These two ATP-driven pumps have similar amino acid sequences and structures, suggesting that they share a common evolutionary origin. Coupled transporters exploit gradients to take up nutrients actively. A gradient of any solute across a membrane, like the Na+ gradient generated by the Na+K+ pump, can be used to fuel the active transport of a second molecule. The downhill movement of the first solute down its gradient provides the energy to drive the uphill transport of the second. The transporters that do this are called coupled transporters. See Figure 12.8. They may couple the movement of one inorganic ion to that of another, the movement of an inorganic ion to that of an organic molecule, or the movement of one organic molecule to that of another. If the transporter moves both solutes in the same direction across the membrane, it is called a symport. Figure 12:16. If it moves them in opposite directions, it is called an antiport. A transporter that ferries only one type of solute across the membrane and is therefore not a coupled transporter is called a uniport. The passive glucose transporter described earlier, see Figure 12:6, is a uniport. In animal cells, an especially important role is played by symports that use the inward flow of Na+ down its steep electrochemical gradient to drive the import of other solutes into the cell. The epithelial cells that line the gut, for example, transfer glucose from the gut across the gut epithelium. If these cells had only the passive uniport glucose transporters, they would release glucose into the gut after a sugar-free meal as freely as they take it up from the gut after a sugar-rich meal. But these epithelial cells also possess a glucose Na+ symport, which they can use to take up glucose from the gut lumen by active transport, even when the concentration of glucose is higher inside the cell than in the gut. Because the electrochemical gradient for Na+ is steep, when Na+ moves into the cell down its gradient, the sugar is, in a sense, dragged into the cell with it. Figure 12:17. Because the binding of Na+ and glucose is cooperative, the binding of one enhances the binding of the other. Both molecules must be present for coupled transport to occur. If the gut epithelial cells had only this symport, however, they could never release glucose for use by the other cells of the body. These cells, therefore, have two types of glucose transporters. In the apical domain of the plasma membrane, which faces the lumen of the gut, they have the glucose Na+ symports. These take up glucose actively, creating a high glucose concentration in the cytosol. In the basal and lateral domains of the plasma membrane, they have the passive glucose uniports, which release the glucose down its concentration gradient for use by other tissues. Figure 12:18. The two types of glucose transporters are kept segregated in their proper domains of the plasma membrane by a diffusion barrier formed by a tight junction around the apex of the cell, which prevents mixing of membrane components between the apical and the basal and lateral domains, as discussed in Chapter 11. See Figure 11:34. Cells in the lining of the gut and in many other organs, such as the kidney, contain a variety of symports in their plasma membrane that are similarly driven by the electrochemical gradient of Na+. Each of these transporters specifically imports a small group of related sugars or amino acids into the cell. But Na+-driven antiports are also important for cell function. For example, the Na+H+ exchanger in the plasma membrane of many animal cells use the downhill influx of Na+ to pump H+ out of the cell and is one of the main devices that animal cells use to control the pH in their cytosol. H+ gradients are used to drive membrane transport in plants, fungi, and bacteria. Plant cells, fungi, including yeasts, and bacteria do not have Na+K+ pumps in their plasma membrane. Instead of an electrochemical gradient of Na+, they rely mainly on an electrochemical gradient of H+ to drive the transport of solutes into the cell. The gradient is created by H+ pumps in the plasma membrane, which pump H+ out of the cell, thus setting out an electrochemical proton gradient, with H+ higher outside than inside. In the process, the H+ pump also creates an acid pH in the medium surrounding the cell. The uptake of many sugars and amino acids into bacterial cells, then, is driven by H+ symports, which use the electrochemical gradient of H+ across the plasma membrane in much the same way that animal cells use the electrochemical gradient of Na+. In some photosynthetic bacteria, The H plus gradient is created by the activity of light-driven H plus pumps such as Bacteriodopsin. See figure 1128. In other bacteria, the gradient is created by the activities of plasma membrane proteins that carry out the final stages of cell respiration that lead to ATP synthesis, as discussed in Chapter 14. But plants, fungi, and many other bacteria set up their H plus gradient by means of ATPases in their plasma membranes that use the energy of ATP hydrolysis to pump H plus out of the cell. These ATPases resemble the Na+, K+ pumps and Ca2+ pumps in mammalian cells discussed earlier. A different type of H+ ATPase is found in the membranes of some intracellular organelles, such as the lysosomes of animal cells and the central vacuole of plant and fungal cells. Their function is to pump H+ out of the cytosol into the organelle, thereby helping to keep the pH of the cytosol neutral and the pH of the interior of the organelle acidic. The acid environment in many organelles is crucial to their function, as we discuss in Chapter 15. Some of the transporters considered in this chapter are shown in Figure 1219 and are listed in Table 12.2. We now turn to the transport of ions through channels and discuss how this ion flow can generate a membrane potential. Ion channels and the membrane potential. In principle, the simplest way to allow a small water molecule to cross from one side of a membrane to the other is to create a hydrophilic channel through which the molecule can pass. Channels perform this function in cell membranes, forming transmembrane aqueous pores that allow the passive movement of small water-soluble molecules into or out of the cell or organelle. A few channels form relatively large pores. Examples are the proteins that form gap junctions between two adjacent cells. See figure 2128. And the porins that form channels in the outer membrane of mitochondria and some bacteria. See figure 1125. But such large permissive channels could lead to disastrous leaks if they directly connected the cytosol of a cell to the extracellular space. Thus, most of the channels in the plasma membrane of animal and plant cells have narrow, highly selective pores. One specialized channel, called Aquaporin, facilitates the flow of water across the plasma membrane. The structure of this protein allows the rapid passage of uncharged water molecules, while prohibiting the movement of ions, including H+. But the bulk of the cell's channel enables the transport of inorganic ions, mainly Na+, K+, Cl-, and Ca2+. It is these ion channels that we discuss next. Ion channels are ion selective and gated. Two important properties distinguish ion channels from simple holes in the membrane. First, they show ion selectivity, permitting some inorganic ions to pass, but not others. Ion selectivity depends on the diameter and shape of the ion channel and on the distribution of the charged amino acids that line it. An ion channel is narrow enough in places to force ions into contact with the channel wall so that only those of appropriate size and charge are able to pass. Figure 1220. Narrow channels, for example, will not pass large ions, and channels with a negatively charged lining will deter negative ions from entering because of the mutual electrostatic repulsion between like charges. In this way, channels have evolved that are selective for just one type of ion, such as Cl- or K+. Movie 12.5. Each ion in aqueous solution is surrounded by a small shell of water molecules, and the ions have to shed most of their associated water molecules in order to pass in single file through the selectivity filter in the narrowest part of the channel. There, Ions make important but very transient contacts with atoms in the amino acids that line the walls of the selectivity filter. See Figure 1220. These precisely positioned atoms allow the channel to discriminate between ions that differ only minutely in size. This step in the transport process also limits the maximum rate of passage of ions through the channel. Thus, as ion concentrations are increased, the flow of ions through a channel at first increases proportionally, but then levels off, saturates at a maximum rate. The second important distinction between simple pores and ion channels is that ion channels are not continuously open. Ion transport would be of no value to the cell if there were no means of controlling the flow and if all of the many thousands of ion channels in the cell membrane were open all of the time. Instead, ion channels open briefly and then close again. Figure 1221. As we discuss later, most ion channels are gated. A specific stimulus triggers them to switch between a closed and an open state by a change in their conformation. Because an open ion channel does not need to undergo conformational changes with each ion it passes, ion channels have a large advantage over transporters with respect to their maximum rate of transport. More than 1 million ions can pass through an open channel each second, which is a rate 1,000 times greater than the fastest rate of transfer known for any transporter. On the other hand, channels cannot couple the ion flow to an energy source to carry out active transport. The function of most ion channels is simply to make the membrane transiently permeable to selected inorganic ions, mainly Na+, K+, Ca2+, or Cl-, allowing this to diffuse rapidly down their electrochemical gradients across the membrane when the channel gates are open. Thanks to active transport by pumps and other transporters, most ion concentrations are far from equilibrium across the membrane. When a channel opens, therefore, ions rush through it. The rush of ions amounts to a pulse of electric charge delivered either into the cell as ions flow in or out of the cell as ions flow out. The ion flow changes the voltage across the membrane potential, thus altering the electrochemical driving forces for transmembrane movements of all the other ions. It also forces other ion channels, which are specifically sensitive to changes in the membrane potential, to open or close in a matter of milliseconds. The resulting flurry of electrical activity can spread rapidly from one region of the cell membrane to another, conveying an electrical signal, as we discuss later in the context of nerve cells. This type of electrical signaling is not restricted to animals. It also occurs in protozoans and plants. Carnivorous plants, such as the Venus flytrap, use electrical signaling to sense and trap insects. Figure 1222. The membrane potential is the basis of all electrical activity in cells, whether they are plant cells, animal cells, or protozoans. Before we discuss how the membrane potential is generated, however, we look at how ion channel activity is measured. Ion channels randomly snap between open and closed states. Measuring changes in electrical current is the main method used to study ion movements and ion channels in living cells. Amazingly, electrical recording techniques have been refined to the point where it is now possible to detect and measure the electric current flowing through a single channel molecule. The procedure for doing this is known as patch clamp recording, and it provides a direct and surprising picture of how individual ion channels behave. In patch clamp recording, a fine glass tube is used as a microelectrode to isolate and make electrical contact with a small area of the membrane at the surface of the cell. Figure 1223. The technique makes it possible to record the activity of ion channels in all sorts of cell types. not only in large nerve cells, which are famous for their electrical activities, but also in cells such as yeasts that are too small for the electrical events in them to be detected by other means. By varying the concentrations of ions in the medium on either side of the membrane patch, one can test which ions will go through its resident channels. With the appropriate electronic circuitry, the voltage across the membrane patch, that is, the membrane potential, can also be set and held clamped at any chosen value, hence the term patch clamp. The ability to expose the membrane to different voltages makes it possible to see how changes in membrane potential affect the opening and closing of the channels in the membrane. With a sufficiently small area of membrane in the patch, sometimes only a single ion channel will be present. Modern electrical instruments are sensitive enough to reveal the ion flow through a single channel, detected as a minute electric current, of the order of 10 to the negative 12 ampere. These currents typically behave in a surprising way, even when conditions are held constant. The currents switch abruptly on and abruptly off again, as though an on-off switch were being jiggled randomly. Figure 1224. This behavior implies that the channel has moving parts and is snapping back and forth between open and closed conformations. See Figure 1221. As this behavior is seen even when conditions are constant, it presumably indicates that the channel is being knocked from one conformation to the other by the random thermal movements of the molecules in its environment. Single-channel recording is one of a very few techniques that can be used to monitor the conformational changes of a single protein molecule. The picture it paints of a jerky piece of machinery subjected to a constant violent buffeting is certain to apply also to other proteins with moving parts. If ion channels randomly snap between open and closed conformations, even when conditions on each side of the membrane are held constant, how can their state be regulated by conditions inside or outside the cell? The answer is that when the appropriate conditions change, the random behavior continues, but with a greatly changed probability. If the altered conditions tend to open the channel, for example, the channel will now spend a much greater proportion of its time in the open conformation, although it will not remain open continuously. See Figure 1224. When an ion channel is open, it is fully open, and when it is closed, it is fully closed. Different types of stimuli influence the opening and closing of ion channels. There are more than 100 types of ion channels, and even simple organisms can possess many different channels. The nematode worm, C. elegans, for example, has genes that encode 68 different but related K+ channels alone. Ion channels differ from one another primarily with respect to their ion selectivity, the type of ions they allow to pass, and gating, the conditions that influence their opening and closing. For a voltage-gated channel, the probability of being open is controlled by the membrane potential. Figure 1225a. For a ligand-gated channel, it is controlled by the binding of some molecule, the ligand, to the channel. Figure 1225b and c. For a stress-gated channel, opening is controlled by a mechanical force applied to the channel. Figure 1225d. The auditory hair cells in the ear are an important example of cells that depend on stress-gated channels. Sound vibrations pull the channels open, causing ions to flow into the hair cells. This sets up an electrical signal that is transmitted from the hair cell to the auditory nerve, which conveys the signal to the brain. Figure 1226. Voltage-gated ion channels respond to the membrane potential. Voltage-gated ion channels play the major role in propagating electrical signals in nerve cells. They are present in many other cells too, including muscle cells, egg cells, protozoans, and even plant cells, where they enable electrical signals to travel from one part of the plant to another, as in the leaf closing response of the mimosa tree. Figure 1227. Voltage-gated ion channels have specialized charged protein domains called voltage sensors that are extremely sensitive to changes in the membrane potential. Changes above a certain threshold value exert sufficient electrical force on these domains to encourage the channel to switch from its closed to its open conformation, or vice versa. A change in the membrane potential does not affect how wide the channel is open, but alters the probability that it will be found in its open conformation. Thus, in a large patch of membrane containing many molecules of the channel protein, one might find that on average 10% of them are open at any instant when the membrane is held at one potential, while 90% are open when it is held at another potential. To appreciate the function of voltage-gated ion channels in a living cell, we have to consider what controls the membrane potential. The simple answer is that ion channels themselves control it, and the opening and closing of these channels is what makes it change. This control loop, from ion channels to membrane potential to ion channels, is fundamental to all electrical signaling in cells. Having seen how the membrane potential can regulate ion channels, we now discuss how ion channels can control the membrane potential. In the last part of the chapter, we consider how this control loop works to propagate signals in nerve cells. Membrane potential is governed by membrane permeability to specific ions. All cells have an electrical potential difference, or membrane potential, across their plasma membrane. To understand how this potential arises, it is helpful to recall some basic principles of electricity. While electricity in metals is carried by electrons, electricity in aqueous solutions is carried by ions, which are either positively cations or negatively anions charged. An ion flows across a cell membrane. An ion flow across a cell membrane is detectable as an electric current, and an accumulation of ions, if not exactly balanced by an accumulation of oppositely charged ions, is detectable as an accumulation of electric charge, or a membrane potential. Figure 1228. To see how the membrane potential is generated and maintained, consider the ion movements into and out of a typical animal cell in an unstimulated resting state. The negative charges on the organic molecules confined within the cell are largely balanced by K+, the predominant positive ion inside the cell. The high intracellular concentration of K+ is in part generated by the Na+, K+ pump, which actively pumps K+ into the cell. This leads to a large concentration difference for K+ across the plasma membrane, with the concentration of K+ being much higher inside the cell than outside. The plasma membrane, however, also contains a set of K+ channels known as K+ leak channels. These channels randomly flicker between open and closed states no matter what the conditions are inside or outside the cell, and when they are open, they allow K+ to move freely. In a resting cell, these are the main ion channels open in the plasma membrane, thus making the resting plasma membrane much more permeable to K+ than to other ions. K+ therefore has a tendency to flow out of the cell through these channels down its steep concentration gradient. But any transfer of positive charge to the exterior leaves behind unbalanced negative charge within the cell, thereby creating an electrical field, or membrane potential, which will oppose any further movement of K+ out of the cell. Within a millisecond or so, an equilibrium condition is established in which the membrane potential is just strong enough to counterbalance the tendency of K+ to move out down its concentration gradient, that is, in which the electrochemical gradient for K+ is 0, even though there is still a much higher concentration of K+ inside the cell than out. Figure 1229. The resting membrane potential is the membrane potential in such steady-state conditions in which the flow of positive and negative ions across the plasma membrane is precisely balanced, so that no further difference in charge accumulates across the membrane. The membrane potential is measured as a voltage difference across the membrane. In animal cells, the resting membrane potential varies between -20 and -200 millivolts, MV, depending on the organism and cell type. It is expressed as a negative value because the interior of the cell is negative with respect to the exterior, as the negative charges inside the cell are in slight excess over the positive charges. The actual value of the resting membrane potential in animal cells is chiefly a reflection of the K+ concentration gradient across the plasma membrane, because at rest, this membrane is chiefly permeable to K+, and K+ is the main positive ion inside the cell. A simple formula called the Nernst Equation, figure 1230, expresses the equilibrium quantitatively and makes it possible to calculate the theoretical resting membrane potential if the ratio of internal to external ion concentrations is known.